Hello, my name is Colin Conwell, and on behalf of my co-author George Alvarez and our collaborators David Mayo, Andre Barbu, and Michael Bice, who've contributed significantly to this work since its submission to VSS, I'd like to welcome you to our poster. Recent research suggests rodent visual cortex is as well predicted by randomly initialized neural networks as by trained ones. A problem for anyone wishing to replicate in mouse brains the kind of success deep neural networks have achieved in the modeling of monkey brains. The question we'll be asking today is simply whether or not this pattern holds for only certain kinds of neural networks, or for the neural network modeling of mouse brain more generally. To do this, our approach is to use a large amount of models, a large-scale neurophysiology dataset, and multiple methods of neural regression. The tools in our toolkit then are as follows. One calcium imaging dataset from the Allen Brain Observatory, cataloging the responses of over 6,611 neurons to 119 natural scenes. 30 models with different architectures, all trained on object recognition from the PyTorch Model Zoo. 24 models with the same architecture trained on different tasks from the Taskonomy project. 1 representational similarity metric, and 2 neural encoding metrics. Together, this barrage of data, models, and methods allows us some vantage on the possibility space of deep neural mouse brain modeling, and to say a bit more reasonably what seems to work and what doesn't. To give you a better sense of our two somewhat different neural comparison methods, I'll take a bit of time to walk you through both. The first is what we call a non-negative least squares representational similarity analysis, or NNLS-RS for short. This method begins with the calculation of a representational dissimilarity matrix from the average neural responses of a given set of neurons to the 119 natural scenes in our stimulus set. This neural RDM we then use as the target RDM in a non-negative least squares regression, wherein the predictors are a set of RDMs calculated from the features of each layer in a given neural network model. Our desired output in this process is a predicted RDM we can then compare more directly to the brain. To get this predicted RDM without overfitting, we use a k-fold cross-validation procedure in which we iteratively hold out a small portion of each RDM and predict that held out portion with the regression fit on everything else. Each fold of the cross-validation procedure adds more and more cells to our predicted RDM until, after all folds have been tested, we have a now fully predicted RDM in which each cell is the production of a prediction rather than a fit. We then take the correlation of this predicted RDM and the brain RDM to produce a model score. Our sparse random projection and ridge regression method begins not with a representational similarity matrix calculated across many neurons, but with the responses of individual cells to the 119 images in the stimulus set. We attempt to predict these responses with a cross-validated ridge regression in which the predictors are sparse random projections of the responses of a given neural network layer to the same 119 images. As before, we then square the correlation of the predicted responses and the actual responses to attain a score. But note that this score represents a score not for the entire model, but for a specific model layer. This leaves us with various options as to how we wish to aggregate these scores to get a sense of how well overall a given model is doing. We thus aggregate our model layer scores in 2 ways. In the first, we take the max score for each model across all model layers. In the second, we pit those max scores directly against other max scores for each individual neuron, seeing for what proportion of neurons a given model attained the highest overall score. So, what does all this give us? Well, the first thing it gives us are rankings, wherein certain models do better at predicting mouse brain than others. I'll first show you what these rankings look like in general, and then show you how we can use them to answer more specific questions about better modeling the mouse brain. Here are the rankings for the 30 different object recognition architectures, according to the various metrics we just discussed. Here are the rankings for the Taskonomy models, parsed also into various data-driven clusters according to how well training on one task transfers to another. Now, to the questions. One of the first, and the one that initially inspired this work, is whether randomly initialized networks really do predict mouse brain just as well as trained ones. And since we tested both a trained and a randomly initialized version of every model in the PyTorch Model Zoo, we can answer this question simply by looking at their relative scores in terms of average variance explained. In the graphs above, separated into 3 facets by metric, green lines demarcate instances wherein an ImageNet trained version of a model outperforms its randomly initialized counterpart. Red lines indicate instances in which the random model outperforms its trained counterpart. As can be seen in these graphs, ImageNet trained models categorically and by a large margin outperform their randomly initialized counterparts. Next, we can ask what made certain architectures better or worse predictors of mouse brain. In the graphs above, the x-axis is one of 4 variables relating to each model: its total number of parameters, its depth or number of layers, its total number of features or activations, or its top 1 accuracy on the ImageNet classification challenge. On the y-axis is variance explained, scaled from 0 to 1 by method so as to align the different ranges thereof. In concert, these correlations tell us that deeper, thinner models with fewer parameters across larger numbers of activations are more predictive of mouse brain. The main takeaway in all of this is that trained models, no matter the task, do much better at predicting the mouse brain than randomly initialized models do. This makes mouse brains, like primate brains, a more valid target for neural network modeling, and a potentially rich source of insight for computational vision science moving forward. Thank you.